Welcome to our latest session on Scientists Answer Your Questions, a series of free webinars that are sponsored by the participants in the Vitamin D Action Project at Grassroots Health. I'm Sharon McDonald, an epidemiologist here at Grassroots Health. Today we have with us Dr. Sarah Booth, who will be presenting on vitamin K and its interaction with vitamin D. Dr. Booth is a senior scientist and director of the Vitamin K Lab at Tufts University, Human Nutrition Research Center on Aging. We received many questions from our participants about this topic, so we are very excited to have Dr. Booth join us today. So Dr. Booth, it's all yours.
Thank you very much, Sharon. Today what I'm going to talk about is vitamin K and its role in calcification. And I think the question that we want to talk about today is vitamin K another nutrient like vitamin D and calcium in terms of considering what we eat in our diet to promote healthy calcification and to prevent abnormal calcification. So how I've organized my talk today is I'm first of all going to give you an overview of what is vitamin K and the different forms, and I'm going to talk about how much you should eat or what is the state of the science in terms of what we believe people should eat in their diet. We're going to talk about how vitamin K regulates calcification, and then we're going to get to the area that many of you are interested in, and that is how does vitamin K interact with vitamin D? And then I will wrap up with the implications and review some of the more frequently asked questions that we receive on this topic. So let's talk about the vitamin K forms. So when we talk about vitamin K, we're not talking about one single chemical. We're actually talking about 13. And they're grouped into several categories. And for the purpose of this talk, we're going to talk about vitamin K1, which is also known as Phylloquinone. And the one that's getting a lot of attention right now is known as vitamin K2 or menaquinones. So the difference between vitamin K1 and vitamin K2 is simply their structure. So all vitamin K forms have this biochemical structure here, and that supports its main function, which we're going to review. And then they differ by how long this side chain is. So they have different chemical structures. This one has a double bond and this doesn't. And then they can also have side chains that only have 4 groupings here, or they could have 13. And when you see vitamin K2 named as a menaquinone 4,7, that just means that there's 4 of these repeating units. So this is a shorter side chain than, for example, menaquinone 7. Now what's really important to know is that first of all, when we talk about vitamin K2, all menaquinones are not the same. So in some ways it's a misnomer to call it just one form, vitamin K2, and it's better when you read the literature to really scrutinize which menaquinone it is. The second important point here is we get most of our vitamin K as phylloquinone. We get it through leafy green vegetables and plant oils. And when you eat phylloquinone in your diet, it's actually converted in your body in tissues that need it into menaquinone 4. So a very important message is that even though you eat vitamin K primarily as vitamin K1, it does convert to menaquinone 4 of vitamin K2 in the body where it's needed. Now, I just told you that phylloquinone or vitamin K1 is present in green leafy vegetables and in plant oils, but we now know that the menaquinones or vitamin K2 are also in our food supply. Now, unfortunately, in the United States, we haven't systematically analyzed it. However, in the Netherlands and in Japan, they have spent considerable more time analyzing foods for the menaquinones. And there's this food called natto, which is a traditional Japanese food that's a fermented soybean, and they are very, very rich. That's 998 micrograms per 100 grams of menaquinone 7, and a lot of the current vitamin K2 supplements are actually menaquinone 7, and this is the richest source of menaquinone 7. But in terms of other foods that you might see in your diet, we see a lot of the cheeses have menaquinone 9 and menaquinone 8, and we also see some of the dairy products having menaquinone 4. So overall, you can divide the vitamin K1 or phylloquinone into plant-based foods, and the menaquinones appear to originate in foods of animal origin. We also get vitamin K in supplements, although overall there's less vitamin K in dietary supplements than other nutrients such as vitamin D. So phylloquinone is globally available as a dietary supplement, as is menaquinone 7. Now in Japan, menaquinone 4, which is the form that phylloquine converts to in our body, it's actually used therapeutically in very, very high doses of 45,000 micrograms per day, not milligrams per day. And then we have combinations of phylloquinone, menaquinone 4, and menaquinone 7. And these are available in supplements at doses considerably higher than in food. Now, one very, very important issue is that we have never studied the potential interactions of phylloquinone with menaquinone 4 and menaquinone 7 when they're taken in the same supplement. And our experience from other nutrients is that there can be interactions. So right now, even though they are available in these combinations of different vitamin K forms, indeed we don't know how that impacts the way your body uses these various forms. So now we're going to talk about how much vitamin K one requires. As you can see from this slide, there's quite a large range in terms of the recommended intakes of vitamin K. Now, when we talk about recommended intakes for vitamin K, we're referring to the recommended intakes of vitamin K1 or phylloquinone. We simply do not have the studies now to determine unique recommended intakes for vitamin K2. So when we refer to vitamin K intakes, we generally refer to them in response to what people consume from the green leafy vegetables and oils. So in the United States, the recommended intake for women is 90 micrograms per day, and for men it's 120. But as you can see, other recommendations, for example, the WHO recommends 55 micrograms per day for women and 65 micrograms per day. And this wide range simply reflects the fact that we do not have the rigorous criteria by which to determine recommendations. In terms of what people eat, the recommended intakes generated in the U.S. are based on the average intakes of adults in the U.S. So most adults, the average is about 90 micrograms per day, which is about half a cup of broccoli, to give you a comparator in terms of the diet. And men eat a little bit, consume a little bit more. Now, the groups that have intakes that are below the recommended intakes are the elderly. Older people tend to consume about between 60 and 70 micrograms per day. Institutionalized elderly, individuals in nursing homes, etc., Alzheimer's patients, these individuals tend to consume well below the recommended intakes. So therefore, the group that probably is at greatest risk of low vitamin K intake are those of the elderly. Now, a question that comes up a great deal about any fat-soluble vitamin is how much is too much? And at this point in time, we don't have any evidence of there being any adverse events or toxicity associated with vitamin K. Having said that, we have very limited experience with high doses of vitamin K. The vitamin K supplements have just emerged on the market, and very few people have studied them systematically. However, we do know among individuals with normal clotting function, the amount of vitamin K, phylloquinone, and MK4 in the supplements does not appear to be associated with any toxicity. Now, I want to emphasize the fact that that's a statement that only pertains to individuals with normal clotting function. If somebody is on an oral anticoagulant, such as warfarin, any vitamin K form that's in a supplement has the capacity to interfere with the efficacy of it. And therefore, any individual who is on an oral anticoagulant like warfarin must confer with their health professional before they start taking vitamin K supplements because it does interfere with its action. And to that point, there is some evidence that menaquinone 7, which is a very common form of vitamin K2 in supplements, it may interfere with the oral anticoagulants at a much lower dose compared to phylloquinone and MK4. And the reason for this is shown in the graph on the right. And what this shows is if somebody is given equal doses of menaquinone 7 or phylloquinone or vitamin K1, here's vitamin K1, what happens is if you measure their vitamin K blood levels, within 6 hours, there's an increase in the vitamin K, and then it pretty well disappears from your blood. And what that means is vitamin K comes into the circulation, and then it disappears from the circulation very quickly. But as you can see from this red line, menaquinone 7 appears in the blood much sooner, there's a much bigger response, and it stays in the blood for a lot longer. So for this reason, for individuals on oral anticoagulants, there's greater concern about the stability of their oral anticoagulants if they take a menaquinone 7 supplement. So what does this all mean? First of all, we don't know right now how much vitamin K is required for optimal health. And that, I know, is a very frustrating conclusion, but what we recommend is right now eat a diet rich in vegetable and plant oils, and you will have more than adequate vitamin K. Now, clearly this does not apply to patients on warfarin. This recommendation is for people who have normal clotting. So what does vitamin K do? So vitamin K is required for certain proteins to function, and I'm going to walk you through this slide to show you how vitamin K works. So here you have your green leafy vegetables, and that provides the source of vitamin K. And through a lot of biochemical reactions, this vitamin K form actually gets converted in your body to an active form. This active form is required to change a certain protein from a non-functioning to a functioning protein. And then vitamin K gets biochemically changed again back to the vitamin K form that you see in the diet. And this cycle occurs many times. So the take-home message here is vitamin K assists certain proteins to be functional. And it does it with a biochemical change from a precursor protein to a functioning protein. Now, when you don't have a lot of vitamin K in the diet, what happens is there's not enough of this active form, and the protein that requires vitamin K doesn't have a biochemical change, and then it's not functioning very well. And that's really an important concept because you still have the protein in your blood, it's just not in a functional form. And that's the entire role of vitamin K, is to transform certain proteins from a non-functional form to a functional form. Now, we talked a little bit about warfarin, but I thought that it would be important for you to understand how warfarin works and why any discussion about vitamin K and calcification relates to the whole warfarin, the role of warfarin in the vitamin K cycle. So again, here's your green that comes in as vitamin K into your body. It's changed to an active form, which converts a clotting protein, because many of the proteins we require for blood clotting requires vitamin K. It converts that clotting protein into a functional clotting protein, so the blood clots, and then it gets recycled again. So what happens with warfarin, also known as Coumadin, is Coumadin actually blocks the ability for vitamin K to be changed to its active form. So that's how Coumadin works. It stops vitamin K from being converted to an active form. The clotting proteins do not get converted to being functional clotting proteins, and by doing this, you're reducing the blood clotting, and that's what warfarin's purpose is. Now, there's this other way in the body that if you give a lot of vitamin K while you're taking Coumadin, it overrides this effect of Coumadin, and vitamin K gets biochemically changed into the active form, and all of a sudden you have clotting again, which is why anybody on warfarin is told to monitor their vitamin K intake. And this is why, particularly if you're talking about dietary supplements, you have to be very careful of taking on Coumadin, because you will override the effect of Coumadin and you can have adverse events such as bleeding. So now we've talked about vitamin K's function, which is to support these certain proteins. Let's talk about some of the roles of vitamin K involving calcification that overlap with vitamin D. So let's talk about vitamin K in bone. Why consider vitamin K in bone health? Well, first of all, we know that when you measure bone for vitamin K, there's a lot of vitamin K in the bone. And the logic is if there's vitamin K in bone, it must serve as some some function. My apologies. Another reason we study vitamin K in bone health is there's a protein, a bone protein called osteocalcin, that requires vitamin K for its function. So this bone protein is one of those proteins that requires vitamin K to be recycled to give the biochemical change so it has function. We also know that vitamin K, when you take a lot of vitamin K out of the diet, vitamin K status changes. It gets very low, and if you give a lot of vitamin K in the diet, the vitamin K status then improves, which means that vitamin K is one of these nutrients that responds very well to manipulation by the diet, and therefore it's a potential modifiable nutrient in terms of bone health. We also know the groups who are vulnerable to problems with bone, such as children and the elderly. Their intakes tend to be much lower. In the UK, they've demonstrated that intakes of vitamin K among children has been declining over the last few decades. So for these reasons, vitamin K is in bone, a protein in bone, requires vitamin K for function. Vitamin K is easily modifiable, and there are subgroups that are low in vitamin K. For this reason, vitamin K has been considered in terms of bone health. So the first studies that came out related to vitamin K1 in bone, and what they found was that the higher the intake of bone in population studies, the lower the risk of hip fracture. And this was shown in the first 3 studies in adults. There have since been at least 2 studies that have not shown this. When we measure vitamin K1 or phylloquinone in the blood or in the diet, what we have seen is in at least 3 studies, the more vitamin K in the diet or the blood, the higher the bone mineral density. Now, this is something that has not been seen in children, but certainly in older adults. So there you have evidence that the preponderance of evidence supports a role for phylloquinone and bones based on the observational studies. Now, there have also been some studies looking at menaquinone 7 or vitamin K intake and bone. And similarly, the higher the menaquinone 7 intake or plasma, the lower the hip fracture and the higher the bone mineral density. And in children, they've looked at bone turnover, which is an indication of how healthy the bone is, but there has not been any evidence in children, at least, to support this relationship between vitamin K and bone. But the preponderance of evidence in these observational studies indicates the higher the menaquinone in the diet, the better the bone health. Now, these are observational studies, and we can't separate out other factors influencing the intakes or the hip fractures. So the only way you can really determine if a nutrient has a role is to do randomized clinical trials. Now, the majority of randomized clinical trials have been done using phylloquinone. And here I've summarized the 6 main trials that have been done in which they have compared groups with vitamin D and calcium in the control group and phylloquinone in the treatment group. And the phylloquinone has been administered in doses of 500 micrograms, 1,000 micrograms, 5,000 micrograms, and as low as 200 micrograms. And with the exception of this Maastricht trial, the Tufts trial, another Maastricht trial, and then 3 others, there have been absolutely no difference over time in these older individuals when they've either been given a vitamin D and calcium supplement or a vitamin D calcium supplement and vitamin K. And I think that this is a very important distinction in these studies. The majority of the studies, with the exception of the Maastricht trial in endurance athletes, all the other trials have looked, you compared phylloquinone plus calcium and vitamin D to calcium and vitamin D. So these studies are looking at the effect of phylloquinone in a vitamin D and calcium replete environment. And I think that's really important to remember. Now, with the menaquinone 4 supplementation studies, there were numerous studies early on that suggested that menaquinone 4 had a protective effect. However, more recently, when they've compared them either to a placebo or to vitamin D and calcium, we have not seen a difference in the effect of menaquinone 4. And please notice this dose is 45 milligrams, which is equivalent to 45,000 micrograms, which is considerably more than the current recommendations of 90 to 120 micrograms. And then the largest study, which came out in 2009, looked at 4,400 postmenopausal women. And what they found is when you give these women who are high risk of fracture MK4, menaquinone 4, and calcium, their fracture risk was no different than if they just received calcium alone. Now, a recent study came out, which is the first of its kind, looking at menaquinone supplementation for 3 years in postmenopausal women. And what they found, I just want you to focus right here on where it says BMD FENE. That means bone mineral density in the femoral neck. So we're looking at the bone mineral density changes in the hip. And over 3 years, you would expect in postmenopausal women a loss of bone mineral density at the hip. And sure enough, you see this in the study. Now, the line with the open circles are the women who received 180 micrograms per day of menaquinone 7. And as you can see, they had less loss than the other group in the closed circles, which represent the women on the placebo. and these were 55 to 65-year-old postmenopausal women. So over 3 years, the Menaquinone 7 provided a small amount of protection. It did not stop the bone loss, but there was less bone loss here in that group than the group that did not receive vitamin K. Now, what's really important is in this study, the placebo group did not get calcium or vitamin D, nor did the Menaquinone 7 group. And so this is a study which has just compared Menaquinone 7 without any calcium and vitamin D. So we have no sense of whether these women were calcium D or vitamin D replete. And all the other studies with the Phylloquinone were done with the calcium and the vitamin D replete. So this is a distinction that we really haven't solved yet, which is that Phylloquinone and Menaquinone supplementation does not appear to reduce bone loss among adults who are calcium and vitamin D replete. And although this Menaquinone 7 may modestly reduce the bone loss, these are women who are not calcium and vitamin D replete. So what we need now are randomized clinical trials to test Menaquinone 7's efficacy in reducing bone loss in calcium and vitamin D replete populations. So overall, the role of vitamin K in bone health is very modest, but there's growing evidence that vitamin K has a protective role against pathological calcification. So we have known for many years that the person who has poor bone density, who has osteoporosis depicted here, see, this is the amount of bone loss per year. So this would be someone in the osteoporotic range. They also have a lot of aortic calcification. So the person with the greatest bone loss per year has the most increase in calcification in their arteries per year. And the person who has very little bone loss over time has very little increase in aortic calcification. And this has been known since the 1940s. And it was originally thought to be a calcium imbalance, that somehow the calcium from the bones was somehow getting out of the bones and ending up in the vessels. And more recently, there has been a lot of controversy about calcium supplements and their risks for heart attacks and heart disease. And again, the assumption is that these calcium pills that people are taking to prevent the bone loss is somehow ending up in the arteries. Well, it makes for a really good story, but it's not based on what we know of the science, and there is no mechanism that would support a simple passive redistribution of calcium from your bones to your arteries. And the amount of calcium we receive in supplements is so small relative to the overall amount of calcium in the body. And what we realize now is that there are many, many biological reasons for this phenomena of people with greatest bone loss also having greater calcification in the arteries. And to orient you to this slide, we've just taken a cross-section of a vessel. So here's where the blood comes through. And this is the intimal layer, and this is the medial layer. And the reason I've got this slide here is not because we're going to go through all these mechanisms, but just to emphasize that each one of these proteins and these factors are involved in a different mechanism, all contributing to vascular calcification. So it is not a simple imbalance of calcium from one location to another. I also want to point out here, this is a vessel, but here we've got processes that are involved in bone formation vascular bone resorption, but we're looking at a vessel. And what does this mean? That there are complex molecular processes that happen in both bone and the vessel. And so the reason people with osteoporosis are at greater risk of vascular calcification is because the abnormal processes, the abnormalities of these complex molecular processes are happening both in the vessels and the bone. It's not simply an imbalance of calcium. So with respect to vitamin K, low vitamin K has been associated with coronary artery disease, associated with calcification in the coronary arteries. It's been involved in it's been associated with diabetes and chronic kidney disease, and also osteoarthritis and aortic valve disease. And all of these conditions are related to abnormal calcification associated with low vitamin K in these various locations. Now, if you recall, we talked about vitamin K and it being recycling and it being required to convert the precursor protein to a functioning protein. And what I want to emphasize here is that vitamin K, again, comes in as the vegetable. It recycles, it converts the protein, and the one we're going to talk about is called matrix GLA protein, which requires the vitamin K to be converted into the functioning form. When we don't have the vitamin K, the calcification protein doesn't function properly. And we have shown this, we have seen this in a mouse model. This was generated by a group in Colombia, that this protein, matrix GLA protein, when the animal doesn't have this protein, and this is a vitamin K dependent protein, so when there's not any function in matrix GLA protein, the animal is smaller, it has abnormal calcification of the cartilage, and its aorta is completely calcified. Now, you don't want the aorta to be calcified, and you don't want your trachea to be calcified, but this is what happens when you don't have functioning matrix GLA protein. So what the hypothesis is that this matrix GLA protein is formed, and in the presence of vitamin K, it converts to that functional form, and the functional form binds to calcium and stops it from progressing. So here's your precursor protein, here's your functional protein. To get from the precursor to the functional, you need vitamin K, and when it's functional, it binds to calcium, so it stops calcium from progressing in your arteries. And this is where vitamin D comes in. So we know that vitamin D at the molecular level can regulate how much matrix GLA protein is produced. So therefore, it controls how much is actually produced, which serves as the amount that the vitamin K can transform into a functional form. The more vitamin D, the more matrix GLA protein is produced, and therefore the more the vitamin K will then transform it to the functional form. So there have been a number of studies that have reported in animals excessively high amounts of the active form of vitamin D causes kidney calcification. And some of these other studies have reported calcification of the arteries. So let me just walk you through this. So 1,25-dihydroxy-D is also known as calcitriol, and this is the active form of vitamin D. And when you give increasing amounts in the diet, you see more calcium deposits. So of 14 mice that are not given calcitriol, you don't see any calcium in the kidneys. Where you go up to 5 micrograms per kilo of diet, you have 9 out of the 11 animals have calcium in their kidneys, and so that's 82%. So this active form of vitamin D in this mouse model causes kidney calcification. Now I want you to just recognize that these are extraordinarily high amounts of the active form of vitamin D. They're excessively high. Now what happens is, if you remember our model, more vitamin D produces more matrix GLA protein, and you need more vitamin K to make it functional. In this study, we didn't add any more vitamin K, so the vitamin D increased the matrix GLA protein, but it didn't have more vitamin K, so the matrix GLA protein that was increasing was not functional. And sure enough, we showed this. Here, when you've got no calcitriol in the diet, you have this amount of this protein. When you have 5 micrograms per kilo of diet, you have more than double the amount of matrix GLA protein. So when you look at the group that has no calcitriol on the diet, you've got 1 part of the non-functional protein to the 1 part functional. In other words, 50% of the total matrix GLA protein isn't functional and 50% is. When you get to the higher doses, what you're finding is almost 2 parts of the matrix GLA protein is non-functional compared to 1 part functional. So you've got double the amount of the protein that's not functional compared to functional. And this would explain why you've got more calcification, because what's happened is you've taken away the vitamin K, so you don't have as much functional form. And therefore, you're going to have more vascular calcification. So how does this relate to humans? Well, the most important thing you have to recognize when you extrapolate from the animal studies to the human studies is that we do not consume vitamin D in the form of calcitriol. So what am I referring to here? Right here in this very complex diagram of how vitamin D is metabolized, here is calcitriol, and it is the active form of vitamin D that is produced in the kidneys. But we don't get vitamin D from there when we consume it in the diet. We get it in these forms. In other words, this is the dietary form, and the body has exquisite ways of when there's too much in the body, they get stored in the fat, they get converted into inactive products. There's lots of feedback loops so that every time we have too much, they get converted into these forms that can be excreted in the body. In other words, you can't extrapolate the diet data from the animal studies to humans when we're talking about different forms. And so when we consume D in very high doses, there are mechanisms in the body by which to metabolize it so they're excreted into forms that will not cause the harm that we've seen in the animal studies. Now, getting back to vitamin K, what they have shown in these animal studies is that when you give animals vitamin K and when you give animals warfarin, which remember blocks the effect of vitamin K, what happens is in 6 weeks taking warfarin, these animals have a lot of aortic calcification. In other words, blocking vitamin K appears to increase calcification in these animals. But when you give these animals warfarin over 12 weeks, and when you give it over 12 weeks, it continues to increase the amount of arterial calcification. But when you give them the warfarin with the low vitamin K, as indicated by the purple bars, there is no difference in the amount of aortic calcification. But when you give high vitamin K to these animals that have got calcification, what it does is it reduces the amount. In other words, high vitamin K can actually reduce arterial calcification caused by warfarin, and there's about a 37% reduction. Now, we have shown this as well in humans, and I'll orient you to this graph. The blue are people who were taking vitamin D and calcium. These were older adults, 60 to 80 years old, and they were given just calcium and vitamin D for 3 years. And the group in the yellow are vitamin D, calcium, and phylloquinone. And what you see is over 3 years, everybody had some increase in calcification in their coronary arteries, but the individuals who received the phylloquinone had less increase in calcification compared to the group that did not receive vitamin K. And this is whether you adjust for how much calcium they had in their at baseline and how much vitamin K they had in diet. The results are the same. In other words, phylloquinone in individuals who have calcification appears to slow down how much more calcification they get. And I just want to emphasize there are no data to indicate that phylloquinone stops calcification from starting. It's just once calcification is started, it slows down the progression. So what does this mean? Vitamin K does appear to control the progression of abnormal calcification. Right now, there's no evidence in humans that a very high vitamin D intake causes increased calcification in humans. And again, with respect to the vitamin K, the current guidance is still to eat a rich diet in vegetable and plant oils to receive the amount of vitamin K you need. The previous study here, the dose was 500 micrograms per day, which is a very large salad with plant oils in it. So these are pretty modest amounts of phylloquinone. So that concludes my overview of vitamin K and calcification and the interaction with vitamin D. So let's switch over to some frequently asked questions I receive about vitamin K and vitamin K and D. So one of the questions that I receive quite frequently is, what is the best form of vitamin K to consume and how much should I consume? Right now, we don't have enough data to support recommendations to promote any one form of vitamin K or another. Most of our research has been done in vitamin K1 or phylloquinone, and definitely phylloquinone is the most common form of vitamin K in our diet and very easy to attain. It's in vegetables and plant oils. There are certain diets and certain food supplies where you're going to have more of the menaquinones, particularly in food supplies that have a lot more dairy products. For some people, this is more difficult to attain vitamin K because they do not consume many dairy products. So I would recommend to consume vitamin K through a varied diet with a lot of emphasis on plant products. In terms of how much one should consume, right now, the current recommendations are fairly consistent with general dietary guidelines, and if one follows the dietary guidelines for Americans about a well-balanced diet, you will easily attain sufficient vitamin K. The evidence for supporting vitamin K supplementation is still very weak, and because we do not have a great deal of experience at potential interactions between vitamin K obtained from supplements and other nutrients, I think that one has to be very careful about taking too many supplements, and if you can attain it through the diet, it would be preferable at this point. So another question I receive is it important to consume vitamin K with vitamin D supplements? Right now, we don't have any evidence, scientific evidence, to support the need for taking supplemental vitamin K if you're taking supplemental vitamin D. At this point, it still seems prudent to obtain your vitamin K from your diet, and particularly as we don't know if there would be adverse interactions between supplements, vitamin K supplements, and vitamin D. And as I illustrated in my talk, the concern about increased calcification with high vitamin D supplements does not seem to be appropriate this time, because the form we obtain vitamin D in supplements is not the form that has been attributed to increased calcification in the animal studies. So finally, what is the right balance between calcium, vitamin D, and vitamin K? Again, I recommend people work with their healthcare physician in terms of their needs for calcium and vitamin D. Certainly in the older population, calcium and vitamin D intakes need to be increased given their very critical roles in bone health, and there's growing evidence of other roles for these nutrients. And in terms of vitamin K, we still do not have any evidence that one needs to increase their vitamin K intake because they're taking calcium and vitamin D supplements. And the evidence strongly supports obtaining vitamin K from a balanced diet at this point. And I think that until we have a greater understanding of the different forms of vitamin K and the impact of combining these different forms of vitamin K in supplements, it would be more prudent to obtain your vitamin K through a healthy diet.
Thank you, Dr. Booth, for your presentation about vitamin K. We have lots of people who are interested in knowing about vitamin K and its association with vitamin D, so we appreciate you sharing your work with us. I also want to thank all the listeners for watching this Scientists Answer Your Questions video session. Let us know what questions you have for our scientists by visiting our website at www.grassrootshealth.net. On our website, you can also watch other Scientists Answer Your Questions videos on the Video tab and find information about how to join the D Action Project.